Greetings to all. Today we are going to see about Citrobacter frundi. So Citrobacter frundi is a gram-negative bacteria. It is a rod shaped with a length of 1 to 5 μm and a diameter of 1 μm. Most cells are motile, but some are not motile. It is a member of Enterobacteriaceae family, which has 15 genome species. Whilst many strains are considered beneficial, some have developed resistance to common antibiotics. In this bacteria, capsule and endospores are absent and it respires as facultative anaerobic. It grows under optimum temperature of 30 degrees Celsius with a pH of 5.1 to 7. Its habitat is soil, water, intestinal tract of animals and in human clinical samples. It was discovered by Workman and Gillen in 1932. So the pathogenicity of Citrobacter frundi. It transmits by direct contact with hospital staff or through contaminated infant formula. It can also directly transmission from mother to infant, and it also transmits through contaminated food or water or through fecal oral route transmission. It has an incubation period of 5 to 7 days. The virulence factor of Citrobacter frundi includes toxins like Shiga-like toxin, heat-stable toxin, A. cholera toxin and B. subunit homolog. Adhesions, endotoxin, hemolysin, siderophores, and virulence island. Pathogenesis. And it includes steps like adhesion and colonization, invasion, toxin formation, biofilm formation, and immune evasion. Citrobacter frundi has an aggressive adherence pattern that can play a role in its pathogenicity. So, Citrobacter frundi adheres to host cell and tissues, establishing a foothold for infection. Then, it invades the host cell using its T3SS to modulate the host cell signaling and immune responses. It produces toxin that damage the host tissue and contributes to disease. It can replicate with human brain microvascular endothelial cells, which may be a factor in its ability to cause meningitis and abscess. And biofilm formation. C. frundi forms biofilm, which are resistant to antimicrobial agents and host immune responses. The bacterium evades the host immune responses using its capsule and other virulence factor to avoid detection and elimination. Citrobacter frundi can produce virulence related genes such as colonization factor antigen, uriases, and other membrane proteins. Clinical manifestation. So, Citrobacter frundi is a common cause of urinary tract infection, particularly in catheterized patients. It can also cause food poisoning, which is a major result in diarrhea. The bacterium can cause sepsis, a life threatening condition characterized by systemic inflammation and organ dysfunction. Citrobacter frundi can cause meningitis, particularly in neonates and immune compromised individuals. The bacterium can infect wounds, particularly in patients with compromised immune system. It can also cause pneumonia, which is particularly in hospitalized patients. So, lab diagnosis of Citrobacter frundi includes firstly microscopic examination. In gram staining, it shows the pink colored and rod shaped bacteria, which results in the gram negative rod. And it is mostly motile, it shows the present in motility test. Colony morphology on culture medium. In blood agar, it shows circular, flat, entire pink to violet colored colonies. And in nutrient agar, the colonies are smooth, convex, moist, and translucent to opaque with the diameter of 2 to 4 mm. They may be in grayish in color. And in chocolate agar, growth was observed as dramatic swarm fronds. And in McConkey agar, pink lactose fermenting colonies were observed. The selective media for Citrobacter frundi is xyloslysin deoxychlorate agar. Colonies are yellow with a yellow center and a yellow precipitation around it. Biochemical test includes the catalase test is positive, oxidase test is negative, urease test is variable, is either a positive or negative. Glucose fermentation test is positive and maltose fermentation test is positive. Indole test is negative. Methyl red test is positive and Vogus Proskauer test is negative. And the citrate utilization test is positive. Starch hydrolysis test is negative and casein hydrolysis test is positive. The selective biochemical test includes malonate utilization test is positive and acid production from D-arabitol test is positive. So the antibiotic therapy and prevention includes aminoglycosides, fluoroquinolones, and carbapenems are currently the treatment of choice for Citrobacter species. For Citrobacter species causing brain infections, a combination of ciprofloxacin and meropenem is often considered the most appropriate therapy. The vaccines are still not available for this bacteria. And prevention includes many preventive measures are there. Some of them are wash your hands from in person to person and maintain good habits, avoid contact with infected animals, and food handlers with poor hygiene can be a source of infection from Citrobacter species. Thank you.